morning, everybody, or good afternoon, or good evening, depending on where you're located. I want to welcome you to our TG118 webinar that we're going to do on RBE and radiation weighting factors. We're doing this for in association with ICRP. We're part of ICRP, and they have agreed to host us, for which we're very grateful. We have two speakers today, and I'm going to sort of tell you how we're going to run this. Number one, we're going to allow our first speaker, Laure Sabatier, to speak. We'll allow for about 10 minutes of questions. After she's done, we'll have our second speaker, Andrzej Wojcik, who will also speak. We'll allow 10 minutes for questions, and then we'll allow for broad questions. Now, the people that are on this, that are on the committee, you are welcome to ask questions by raising your hand, and I'll call upon you, and it'll allow for a little bit more interaction with the speakers. Those of you that are not members of the committee but want to ask questions, please use the Q&A function, and I will call upon questions in the Q&A as we have time and as we progress through this. And with that, I think I'm going to just get started. I'm not going to do heavy introductions on these speakers because, to be honest with you, we could take up the entire time introducing them. Our first speaker is Dr. Laure Sabatier. She's at the Atomic Energy and Alternative Energy Commission. She's research director at the CEA. She has numerous publications, I think key publications in the field, and it was some of the work that she's been doing on sort of alternates to RBE that made us be interested in her work. Laure, please, why don't you start?
Okay, perfect. So first, I will talk about the relative dose effect as a ratio of effect for a given dose. And we think that it allows a simple comparison of response curves. So it's in fact what we prepare when we prepare this work. It's a rather long time ago. It has been published in 2016, and I really want to thank Gail to ask me to talk about it, because frankly, for me, it was really a long time ago. And as you know, there is many things that could occur within 5 years. So what we have done in that time, and the wish was to compare individual radiosensitivity to gamma rays and carbon ions. And meanwhile, we identified that RBE was not suitable for us, as our dose response curves due to individual radiosensitivity was, to our knowledge, not powerful enough. And we were more efficient in comparing isodose effects. So we propose and we introduce a new term that we call relative dose effect, which permit to compare the biological damage that we chose to study at a single dose. So what so that is a curve that you know very well, which is a comparison of two kinds of radiation by scoring dicentrics and induced by low or high LETs. And I like this curve because we could see both in the square the RBE as a ratio of those that we will produce an ISO effect, and RDE, where for a single dose we could compare the ratio of effect induced by a single dose. So our main purpose when we start the studies is to compare two kinds of radiotherapy. So the conventional radiotherapy, low LET, photon beams with 2 gray per fraction. and the fact that adjuvant therapy was increasing, and charged particle, and mainly carbon. So the question that medical doctors ask us is, what is the effect of 2 gray exposure? If I give a fraction of 2 gray, what will be the magnitude of the differences between carbon and photon? So we decide to study many donors and to select the biological effect that would be for us the most robust. So this cartoon is only to summarize the fact that radiotherapy is shifting to the use of heavier ion species. Up to now, carbon ion therapy is not yet widely used, but it's really increasing. And between 2016, when we published this paper, and now it's really increased a lot. So the question that we ask is, first, is individual radiosensitivity to one type of ionizing radiation is correlated to the radiosensitivity to the other type? And what is the RDE of carbon ions compared to photons? So we decide to score chromosome aberration, and we use a technique which modify a little bit the gold standard, which is a scoring of chromosome aberration after uniform staining and GEMSA staining. And we use both telomere and centromere labeling, and to reduce ambiguity in the counting of dicentric and the centric rings. Just a few examples of ambiguity that we could have. For example, here we could hesitate, is it really a dicentric, or is it only a break at the very end? And here it's clearly a dicentric with the 2 green spots. Here we could really distinguish that is the 2 chromosomes and not a dicentric. And here we could really identify that it's not a dicentric, even if it was tempting to say that it was, but it's a normal chromosomes. And more tricky in this one, we could see that here, what we could think it's a dicentric, in fact, is a tricentric with 3 centromeres. So it's permit to optimize the dose effect curve that we have in the lab. Here it's a dicentric per cell, and that is a classical curve in green. That is in red what we obtain with solid GEMSA staining, and in blue the improvement that we have scoring dicentrics with telomere and centromere labeling, and it's even more clear at low dose below 1 gray. So, but now we want to compare low LET versus high LET. And if Gail asked me that I present the paper 2016 paper, really induce coming back to the past. I really had to exhumate other papers when we start this work. When we decide to compare many years ago different type of heavy ions, the first studies that we had done, I remind you, it was in the late 80s. And it was before trying to quantify the damage induced by any kind of radiation. We need to qualify if the damage induced are similar. And what we observe and what we published at that time is the fact that the single traversal of heavy ions, here it's a single traversal of neon with of 200 MeV. And we could observe that we have the induction of complex rearrangements. So those complex rearrangements, let's just guess that it's solid DMSA staining. You will see, for example, here that you have one dicentric and another dicentric. But if you look a little bit more, in fact, that is a four-guy circle with this dicentric will come from 7 and 11, but the piece of 7 is fused with the chromosome 2, who form a dicentric with a chromosome 4, and the piece of chromosome 4 will fuse with chromosome 11. And the remaining part of 11 is fused with 7. So, but in that case, if we compare what we observe between the number of dicentrics, 2 dicentrics, a number of breakpoints, misrepair, giving those dicentrics, that means 4 breakpoints, exactly the same as what we have when we perform this painful air banding. But in some cases, it's not so easy. Look at that, this complex rearrangement where we have only 1 dicentric that could be detected by a solid DMSA, but in fact, and so that would mean 2 breakpoints, but in fact, we have a complex rearrangement with 6 breakpoints with several acentrics. So that is what we observe, and we observe the increase of complex rearrangement according to LETs. When we start the experiment with carbon ions, we perform the study with MFISH, and we look at the mean number of chromosome abnormality, looking at translocation and dicentric or reciprocal translocation, deletion, loss, and addition. And what we observe is that the carbon irradiation induced 3 more non-reciprocal translocation and 6 more deletion. The fact that such kind of heavy ions induce more complex rearrangements, more deletion, has been published by several teams, and it let us ask how we will compare different kind of radiation, and the fact that if we look only at dicentric, like in the gold standard, we will miss a lot of damage. If we look at only deletion, it will be the same. So we decide to have a common measurement, which will be the number of breaks that are unrepaired or misrepaired, forming chromosome rearrangements. And when we perform such kind of counting with telomere and centromere labeling, so we have dicentric, 2 breakpoints, tricentrics, 4 breakpoints, and rings, 2 acentric rings, and we also could distinguish different type of acentric according to the number of telomere, and we could know that this one come from 2 breakpoints, this one from 1, and this one from 2. If we look again to the dose response curve, those has been established with 16 donors, healthy donors, you could see, and you look at the number of what we call abusively DSB, but in fact it's a misrepair DSB giving chromosome rearrangement. We could see both how we could calculate it with DMSA staining and those with telomere and centromere staining, and how more efficient it is. So now coming back to what we observe in our comparison of gamma and carbon. So we have a set of more than 10 donors, 18 for gamma and 13 for the carbon. And first, what we observe after 2 gray irradiation is the fact that they are not responding by the same way. And if we rank them from what we call the most radioresistant to the most radiosensitive, we could see that even if we observe something like around 2 DSB per cells, misrepair, we have a range of 1.84 differences between some donors. And these differences is even more explicit when we look at the number of DSB per cells, and we find few donors with a very large number of DSB per cells. What we observe with carbon, so it's a carbon that we perform in Ganil, in Caen, and it's carbon 75 MeV, and we select those carbon with these LETs because this LET at the plateau region of the Bragg curves is the same LET that we have in the Bragg peak of the 400 MeV carbon, who is a carbon used for adjuvant therapy. What we observe with carbon, we observe that again, we had for carbon, we had only 13 out of 18 donors because of the amount of blood available. And those 13 donors, again, we have a large differences among donors between for the same 2 gray, and those donors has a large differences in the number of breaks per cells. We try to compare both the radiosensitivity between gamma rays and carbon. Here, it's maybe more evident when you look at here. We look at the radiosensitivity with gamma and with carbon, and you could see that there is absolutely no correlation between the radiosensitivities that we observe with after gamma and with carbon. So we conclude that it's very important to have a large number of donors when we perform those experiments, and to calculate the mean of the DSB per cells for a large number. So the number is very important to reduce the effect of the individual radiosensitivity when we want to answer to the question how carbon is more efficient than gamma for a 2 gray fraction. As we were not able to perform all those experiments for many donors with a very large range of dose, and how the question of the medical doctors was about 2 gray, we decided to focus on the 2 gray and to calculate the RDE and not the RB. So it's as simple as this, even if it could seem rather provocative to use RDE instead of RB. We just check one thing that we check with 4 donors is to see if the fact that we use telomere and centromere staining instead of using a technique that permits to identify any type of chromosome rearrangement, if it will modify the RDE that we observe. So we calculate only for 4 donors. We analyze by exactly the same metaphase labeled with telomere and centromere or labeled with MFISH, and we looked at the amount of DSB per cell that we could score. Of course, after MFISH, we score far more chromosome rearrangement, so far more breakpoints. 2.2 versus 3.3 for gamma, and 6.5 versus 9.8 for carbon. But when we look at the RDE that we observe, we exactly observe the same RDE independently of the techniques that we use. So we were a little bit happy with this type of results, and we focus on 2 gray by analyzing as much as possible donors that we have to be able to answer. So maybe in this cartoon, I summarize the comparison of individual radiosensitivities that we observe at a dose of 2 gray. So we observe the same range of differences in radiosensitivity, 1.7, 1.6 fold between for carbon and for gamma. And the RD that we observe between those donors, 18 in that case, 13 in that case, is 3. But we then address the question, what will occur for the few donors for which we were able to perform those effect curves? And could we calculate the RD in that case? So here we perform for 6 donors for gamma rays and 4 donors for the carbon. We prepare those effect curves that you have here, and we could see we have a little bit more dose with high doses, but in that case, you could see that the error bar are really large, and we are not very confident in what we observe at 10 and 15 gray. So what we observe concerning the RDE, you see that first, RB is still here because first I tried to have on the same curve RBE and RDE, but it was rather confusing. But so this one is only the RD part, and you could see that here it's 3, but it decreases with dose. So it's very important when the medical doctor asks us what they want to know is the dose per fraction. And for them, the fraction is 2 gray. If we go below 2 gray, is hyperfractionation. If we go above 2 gray, it's hypofractionation. So we could see that a little bit of hyperfractionation will be useful in that case, but when we present this, they thought that 2 gray was a very good, interesting differences between photon and carbon. So the conclusion is first, it's very important to select the biological effect, and scoring all chromosome breaks help us to be able to compare different type of radiation and to homogenize the variation in the quality of chromosome aberration, which are induced by different type of radiation. RDE for us was not, when we proposed it, it was not to be provocative, but it's only to have a useful tool for a simple message for the medical doctors. And to be able to say that a 2 gray carbon will have 3 times more efficient than gamma to induce chromosome breaks. So now I want to thank Gail to ask me to present this data, because now I'm a little bit excited for those results that I have a little bit put in the back of my mind after all those years. And I would like to see how to use that tool for single track traversal and mixed beam for all the work that we perform during all the old time with all those heavy ion irradiations that we have scored for space exposure, but it will be another story. So thank you very much.
Laure, thank you. This was really great. Really enjoy it. We're now open for questions. And first I'll take from the panel, and then I'll take from others. There are quite a few questions that have popped up. We will take only 10 minutes for questions now. And then if we don't get to it now, we'll hopefully get to it later. The questions will accumulate. I think Andrzej had his hand up, but maybe I'll start with the first quick question. Question number 1 is kind of easy-ish. Did you ever see any evidence of chromothripsis when you were doing your chromosomal studies?
Not with carbon, not with carbon. Chromothripsis we could see with some heavy ions with a very high helices but in this range of dose between gamma and carbon, we have not seen it.
Okay, and my second question is, have you ever tried this in animal, most of what you're doing is either in vitro or ex vivo. Have you ever done anything in vivo, like in animals?
No, never.
Yeah, that would be cool.
Mainly because we perform most of our experiment in Ganil, and in Ganil, it was not allowed to expose the animals. So that's mainly why we have not done it.
Great, thank you. Are there any questions from the panel first? I mean, Andrzej, I saw your hand up, but then it went down. And does anybody go ahead?
Because I actually wanted to clap hands, but I couldn't. I couldn't find. But I can ask a question. Okay, Laure, thank you very much. That was very nice, very interesting. But the question from the perspective of our task group on RBE, when you calculate RDE versus RBE, which value is higher? Or did you play around?
Yes, it's always RDE. For example, we have an RBE of, if we look at the effect at 2 gray, and we calculate the RBE, and we have 2.6 for the RBE instead of 3.
Okay, thanks.
Are there any other questions from the panel before I go to the open questions? I'm not seeing any, so I'm going to go to the open questions now. Let me start. So this is from Tatsuhiko. The advantage of RBE includes its use as a dose conversion factor. That is, dose of low LET radiation equals RBE times dose of high LET radiation. Thus, the radiation weighting factor is based on RBE. Is it practical that we stop, that we totally stop using RBE?
I don't think so. I think that when you have a very good dose effect curve, RBE is very useful. But the problem is that there is 2 problems. Is that when we are looking at biological factors, where we have a large individual variation, it's very important to have a large number of donors. And sometimes it's not as we have, and it was our case. It was not possible to use a very large number of donors for the full range of those response that could permit to calculate the RBE. So to be able, I think it's more efficient to have a lot of donors and to look at the dose in which medical doctors are interested in, than to have few donors, large dose response, and to calculate the RBE to be able to give an indirect response to the effect of what happened at 2 gray. So it really depends of who addressed the question. In our case, it was the medical doctors, and what is the individual variation in the biological effect that you are looking at.
Great, thanks, Laure. Here's another question. Does radiosensitivity also depend on the energy of the beam?
We don't have, I'm not able to answer because I test only the carbon. And when I look at the energy of the beam for neon, for example, I always add the same donors. So I try to look back in all data that I add if I could address this question, but I never add enough donors to observe the variation. I always add 2, 3 donors because at that time I thought that 3 donors was enough to calculate the mean. And it's only more recently that we decide to increase the panel.
Okay, great. Here's another. Is it possible that the difference in double-strand breaks are related to mass collisions versus electromagnetic interactions, such as Compton scattering interactions with the gamma radiation?
I think that here, we are not talking about the double strand breaks which are induced at the very beginning, but as those which are misrepaired and giving chromosome aberration. So I don't think that I think it's more linked to the energy deposition and the fact that we have a lot of energy deposition really nearby, and to have more complex breaks than the differences which is addressed in the question.
Okay, this is going to be the final question that we'll take before we move on to our second speaker. Is the difference in radiosensitivity of donors repeatable? That is, are there intrinsically radio-resistant individuals? And then a second part to that question, similarly, does the radiosensitivity correlate with treatment outcome?
It's not, that is healthy donors. They come from the blood bank. So I don't know if they are in vivo resistant or radiosensitive. When we perform the experiment, for some of them, we perform 3 times, and we have always the same kind of radiosensitivity or radioresistance for a type of radiation. But that is only 3 times that we have done it.
Great, thanks, Laure. Now we have a few more questions for you, but what I'd like to do is have Andrzej talk next, and then what we'll do is come back and ask questions for both you and Andrzej again, but we'll let Andrzej have some specific questions and then we'll go from there. So let me introduce Andrzej again. I'm not going to go into the details of his life. He's amazing. He is the head of the Center for Radiation Protection Research in the Department of Molecular Sciences at the Wenner-Gren Institute at Stockholm University. He's a member of our TG118. So therefore, we're extremely excited to have Andrzej present to us today.
Thank you very much, Gail. Yes, thanks for this opportunity. It's a great event, I think, and I look forward to the discussion. What I would like to show you today are some of our results, but also, what should I say, my lack of open questions that I have that kind of buzz through my head, which I don't know how to tackle, how to answer. And it's about the problem of RBE, of course, because this is a TG118 meeting, and it relates to 2 things: the effect of combined exposures of low and high LET radiation, and also it relates to the problem touched already by Laure, and that is, how can we infer RBE if we don't have a dose response? Okay, let's start with it. It will be mainly, or a lot will be about mixed beams. So what happens? The question is that we are interested in, that we do research on since quite a number of years, is what happens when you expose cells to high and low LET radiation. So they experience this complex damage at the same time as they experience the scattered damage from the complex damage for alpha particles and the scattered damage from X-rays. And again, I will show you some results, but it's really brainstorming. We want to, because we are now in the process of revising the recommendations, and we need input. We need to brainstorm and see, well, should we change anything with respect? Of course, at the end of the day, the radiation, the RBE values that we derive are interesting because of the radiation weighting factors for stochastic effects to calculate the effective dose. So let's focus now again on mixed beams. There is quite a lot of data on the effects. I mean, there is a lot of data on the effects of low LET radiation and the effects of high LET radiation. We know that high LET radiation induces DNA damage of high complexity. There is a high RB, and the ICRP uses the radiation weighting factor of 20. For low LET radiation X-rays, it's of course the other way around. I mean, RB is low and weighting factor is 1. Why are we interested in mixed beams? Because in fact, in very many cases, we are exposed to combined to a mixed beam of radiation of different qualities. So if you're at home and you have high radon, you're also exposed to gamma. If you fly in an aeroplane, you will be exposed to really complicated field. And especially if you're an astronaut, even more so. Cancer patients that are treated with IMRT, you have gamma radiation, you have neutrons. And of course, if you treat patients with boron neutron cancer therapy, it becomes extremely complicated. The radiation field is extremely complicated. And again, there is very little data on what happens in cells that are exposed under experimental conditions. I think the problem is that there are few people that have radiation exposure facilities that allow you to expose cells simultaneously to high and low LT radiation. As you will see, we built such a facility, and the results that I will show you are from this facility. So what do we expect? Yeah, well, what can you expect? The easiest thing is it doesn't matter for the cell whether it's exposed to low and high LT radiation in terms of a possible interaction. There is no interaction. Okay, you're exposed to low LT radiation, at the same time high LT radiation, fine, low LT radiation is repaired, high LT radiation damage is repaired. The effects are additive. This is what we assume in ICRP, that there is no synergistic, there is no interaction. But it is possible that DNA damage complexity increases, and the effect would be synergistic, that the high LT exposure leads to opening of chromatin, and you get more oxidative damage, then you would get synergism. Maybe the high LT exposure overwhelms DNA repair. The DNA repair focuses on the complex damage, and then the low LT damage is not repaired properly. Then you would have a synergistic effect as well. So what's the RBE of high LT radiation under mixed exposure scenario? And the question is also, it's not just academic, because if you look at the LSS results, which are really the gold standard for our risk assessment, there people are exposed to a mixed beam of neutrons and gamma radiation. And we know that the radiation awakening factor for neutrons, despite the fact that the neutrons were of different energies, we just take it for granted it's 10 for simplicity reasons. But we don't assume that there is any, or we assume there is no synergism, there is additivity. So here is the facility that we built. It's here you have an alpha exposure facility. You don't alpha exposures and as americium-241 source. You don't see it. It's at the bottom of this dish. We spread cells on a disc like this, covered with a mylar foil. Here it is blood. You push it against the source, and at the bottom you have an X-ray machine. So you can expose cells to alphas, to X-rays, or at the same time to both radiation types. Yeah, well, how do we evaluate whether you have synergism or additivity? You calculate something that is called envelopes of additivity. It's one of the techniques. I will not go into detail more about this, but you will see these envelopes of additivity, and if your found observed effect is to the left of the envelope, it means synergism. If it's inside the envelope, it's additivity. If it's to the right, is sub-additivity. And we have done a number of experiments with different endpoints, micronuclei, looking complex chromosomal aberrations in lymphocytes, DNA repair kinetics in different cell types. I will focus on the most recent stuff, and that is formation and decay and movement of DNA repair foci in U2OS cells, gene expression in human peripheral blood lymphocytes, chromosomal fragments in human peripheral blood lymphocytes, and then effects after low dose rate exposure. So let's see at 53 BP1 foci, we can see them. We have cells, U2OS cells that are transfected with a plasmid that code for 53 BP1 with gene green fluorescent protein. So we can look, we don't need to go with antibodies. We can see the foci live, but we did first experiments where we fix the cells and then we look at the foci. Here is the dose response. So here's the frequency of foci per cell as a function of dose, alpha particles, X-rays, and mixed beam. We can differentiate between small foci, we assume reflect simple damage, and large foci, which we assume reflect complex damage. And you can already see that if there would have been additivity, the mixed beam dose response curve should be somewhere between the X-ray and the alpha particle dose response curve. But we can apply our envelopes of additivity, And if we do this for small foci or for large foci, sure enough, we always see, this is calculated for different levels of frequency. So small foci frequency 5 per cell, 10 per cell, 13 per cell, and large foci frequency, it's always there. You always see synergism. So there is an interaction. We can also look at foci life. We have a life chamber cell, or yeah, it's a microscope coupled with a chamber where we expose cells, and then we look at the formation of foci life. And what we observe is that there is a difference in the way how these foci move around the cell. It's one of the results. I will not show you all the results, but one effect is that what we call mean square displacement is low for mixed beams. So this is mixed beams, and here is the value of the mean square displacement. It means that under mixed beam exposure scenario, the foci do not move as much as they move when they are exposed to alpha alone, to X-ray alone, or in the control cells. What impact does this have on DNA damage repair, misrepair? I don't know, but well, perhaps the repair is not as good. Let's see. Now the next set of experiments that I want to show you, or set of results, is on the expression of the FDXR gene in human peripheral blood lymphocytes, analyzed by RT-PCR. The FDXR gene is known to be extremely radio responsive. So it's used for the purpose of biological dosimetry. And we took two donors, donor 3 and donor 4. Their numbers are for historical reasons. And similarly, as Laure, we are kind of obsessed with the idea of determining individual radiosensitivity. It's just that we didn't look at many donors, but we took these 2 donors and we were collecting blood throughout the whole year. So 3 times 3 repeats. We collected blood in the winter, in the spring, and in the autumn. So once every week during 1 month, then let's say in the winter, then during springtime, every week 3 times, and then during summer or autumn, now I don't remember, again, once per week during the consecutive 3 weeks. And we looked at the expression of the XR gene, and here you see the dose response. It's curvilinear, it saturates at 2 gray, but you see that mixed beams for this donor, mixed beams are above alphas and X-rays are here. And for this donor, it looks rather similar. And if you calculate the envelopes of additivity, again, we see a synergistic effect of both radiation types. Then we also looked at chromosomal aberrations in the same donor. So you run the experiment, you collect blood, you analyze gene expression, and then we also analyze chromosomal aberrations. The problem is we often, because of the alpha radiation exposure, we added calyculin A to induce PCC, premature chromosome condensation. The quality of the slides is not so good. So that's why we are analyzing fragments and not dicentric chromosomes, for example, here, because we can't really identify dicentric chromosomes always. So we focus on fragments just to explain why you see here the result expressed as chromosomal fragments per 100 cells. Again, donor number 3, we didn't analyze all results yet. So this is from 1 set of repeats, 1 times 3 repeats, donor 3, donor 4. But you see a similar effect. Now here we don't need to apply envelopes of additivity because the dose response is linear, at least we can fit it linearly. And here you have X-rays, here you have alpha particles, the red line, and the green line is mixed beams. Donor 4 shows slightly fewer aberrations, but the effect is there as well. The effect, I mean, the combined exposure effect, meaning that the observed mixed beam-induced aberrations are above what you would expect. And the expectation is calculated, the expected value is calculated based on additivity. So we can calculate the RBE of alpha particles here. That would be 4.1. For this guy, it would be 2.9. And we can also calculate the RBE of mixed beams. And here it would be 2. The RBE for mixed beams is calculated as the dose of expected effect versus the dose of the observed effect. So we have, for this donor, under mixed beam exposure scenario, the efficiency of or the effectiveness of alpha particles and of the mixed beam is twice what you would expect. And here it's about 1.5. Okay, so it seems that under mixed beam exposure scenarios that there is synergies. So how do we consider this in radiological protection? Should we consider this? That's one of the questions. But then, of course, one thing that we should bear in mind is that these results were generated with the exposure facility that I showed to you, where we expose cells acutely. So the duration of exposure is minutes. But and most of the data that we have, not just we, but that data that is available from which we calculate RBE, is from such experiments where you expose cells acutely, you generate those responses, you calculate the RBE, and then you apply the RBEs to calculate, or you base the radiation weighting factors, and then you apply these radiation weighting factors, you calculate the effective dose, and you apply this to not the effective dose maybe, but you infer the risk from, or you calculate the risk for chronic low dose exposure. So we thought, well, let's have a look at how do cells behave when we expose them chronically to mixed beams. And for this we built a new radiation exposure facility, which, well, we used an existing radiation exposure facility where we can grow cells in an incubator that is positioned above a cesium-137 source. But then we ordered these little americium-241 sources that have an activity of 200 becquerel, and then you grow cells on mylar discs or dishes that you place directly on these alpha sources. And then you get a dose rate of about 1 milligray per hour. And again, you can grow cells and look on the dishes in a normal incubator. You can place this whole thing inside this incubator and turn on the gamma source, so you will have a mixed beam exposure scenario, or you can just grow your cells inside this incubator without an alpha source, and then you have pure gamma radiation. And well, what do we see? We don't get a mixed beam effect. We looked at least for the moment, we have only results for gene expression, and we lose 2 cell lines, VH10 cells, human fibroblasts, and human lymphoblastoid cell line AH1. And the 200 milligray exposure corresponds to approximately 1 week of exposure. We analyze different radio-responsive genes, among others FDXR. If you pull all the genes together, this is what you get for VH10 cells. You see there seems to be a peak of activity of gene activation for alpha particles, but not so for mixed beams, which is strange because the mixed beam, it contains alpha particles. So where is the effect? It's like canceled out. And we don't see any gene, any response at the gene expression level for H1 cells, which is surprising because these cells are lymphoblastoid cells, which are generally, I mean, we know they are more radiosensitive in terms of their of the killing effect of radiation. Still, we don't see an effect at the level of gene expression. And I don't know why. I don't know how. We can't explain it right now. We are looking at other endpoints. We are scoring micronuclei. We're looking at apoptosis. We're looking at senescence, and so on. No results yet. But I show this to show you that, or what should I say? Yeah, I show it to show that things are complicated. And it's maybe one has to be careful when you extrapolate results from acute exposure to chronic exposure. I mean, we know this, but again, this is yet another reminder. Careful. Okay, let's go back to foci, and let's go back to the problem of RBE, of alpha particles and mixed beams for foci. Now, here you see that the dose response for alpha particles is lower than that for X-rays. So actually the RBE is below 1. And this is not a unique result. There are other results suggesting that the RBE is either 1 or lower than 1. This is from a publication by Franken et al. So what do we do now? How do we consider these results? Do we just say, okay, we don't use it at all for RBE, for calculating, for estimating radiation weighting factors? Okay, just disregard these results? Yeah, well, let's have a look at, I mean, what I showed you here is the immediate dose response effect. So you expose cells and then half an hour later, you look at foci formation where you have the peak of foci. But let's follow the fate of foci. How do they decay? How do they form and how do they decay? So here you have one dose and different times after exposure in hours. So we see this typical peak in foci formation, like 30 minutes after exposure, then a decline, and then, yes, what then? For X-rays and for mixed beams, we had more foci than for alpha particles. Then you have a steep decline. For alpha particles, there was a peak, then there doesn't seem to be a very strong decline. You can see this much better if we fit the data, if we fit the results. So here again, and it's not just that we fit the results, we also normalize them to the frequency of foci after half an hour. So this is now, the results are expressed as % of the maximal focus number, which is after 30 minutes. So here, alpha particles, X-rays, and mixed beams all start with 100%. And now you can see that X-ray induced foci decline very quickly or fairly quickly. Mixed beam foci, and these are now these ones here. This dashed line, first they don't decline, they decline with a delay, but then they decline. But alpha particle induced foci, they kind of remain, which is not surprising because we know that alpha particles induce many complex lesions that are difficult for the cells to repair. Fine, so we don't, if you look at the dose response, back to this slide, you get an RB of lower than 1, but if you follow the DNA repair, you see a clear higher efficiency, detrimental effect of alpha radiation. How do we consider this? Can we calculate something that maybe we could call biological effectiveness factor? In this case, it would be 2. Can we infer RBE from the biological effectiveness factor? Does it at all make sense? I don't know. This is also one of the questions. Should we consider this in our similar results? I mean, I'm just showing you results from our lab, but there are many more results out there, which I didn't systematically analyze yet, but maybe we come up with biological effectiveness factors which are higher than RBE for alphas. Should we do this? Does it at all make sense? It's not RBE, but these are factors that of course play a role in the biological effects of alpha radiation. So if we come to the end now and look at what is known as the hallmarks of cancer, and I believe these images like these are kind of shown much too often, perhaps, but this is a very recent update of the hallmarks of cancer published by Hannah alone, where you have a list of all the possible factors that contribute and that can modulate the risk of radiogenic cancer. We know that radiogenic cancer is induced by DNA damage and induction of genome instability, mutations. But then you have all these factors that can modulate the risk. And maybe these factors, many of them will probably not show a dose response, but maybe they will be sensitive to radiation quality. Should we consider them? How do we consider them? So the questions that I want to ask up for discussion: should mixed beam and combined exposure effects be accounted for in terms of RBE for deterministic effects or radiation weighting factors for stochastic effects? How far can results from acute exposure be used to predict effects after chronic exposure in terms of RBE calculations? Can we infer, should we infer RBE from results where no dose response relationship exists? So do we just say, well, there is no dose response, we can't do anything, we just disregard this effect? How do we call it then? If we do try and infer the RB from such results. How do we call this? Biological effectiveness factor, like BEF, maybe? Then maybe I'm complicating things too much. Maybe it's not necessary. Perhaps we have the radiation weighting factor of 20 for alpha particles. Maybe this is sufficient. Maybe if you look at this, I see. Oh no, yeah, okay. This maybe you can see it. I mean, it's there is an ICRP document number 92 on RBE, and there, for example, they say, well, we know that when they debate the problem of reference radiation, and here we know that gamma radiation and X-rays have a weighting factor of 1. Of course, we know that low energy X-rays have an RB of 1, 2, 3, but in the report, the author says, well, it doesn't matter. It's negligible. So perhaps here the mixed beam effect also is negligible. Maybe we just don't need to worry about this. I don't know. This is something that we need to discuss in TG118, but maybe also you can come up with suggestions or with your thoughts. These are the people that did the work, mainly PhD students, Elina, Alice, Mila, that is now carrying out her results. In fact, my colleague Lovisa, Adriana, who is also a PhD student, and Beata, who is a researcher at Warsaw University. Okay, thank you very much.
Thanks, Andrzej. This was really great. Both talks were very provocative and make us think about things we haven't been thinking about with RBE. One of the questions, I'll start with a question, and then I'll also ask the committee if you have a question to raise your hand. Have you done like proportions of mixed beam, like more of one, more alpha than have you tried to mix that? Because I think that might be a variable here too, right?
Yes, we have some results on this, but they are not really that conclusive. The problem is that these results take a lot of time, so it's not so easily done. What we have also been doing is, but we haven't published this yet, and is to try to irradiate cells, in succession. So first alphas, then X-rays, or the other way around. This is actually stuff that Adriana has done, but we are now looking at the results. And so I can't answer the question yet whether there is an effect of does it play a role, and what is the time span between the irradiations? Does it have to be simultaneously? Or, I don't know. But of course, this is a valid question.
And the other question I wanted to ask is related to that, like moving around of the foci that you talked about early on. Do you have any idea what the mechanism might be that's causing the mixed beam to have a different result than the others? I mean, because that would actually be important to us, right?
Yeah. Well, again, Adriana has been doing this, analyzing this with now recently, but we don't have we are actually kind of evaluating the results. So, but a simple explanation could be that you get these foci exposed or induced by low LET radiation that are scattered all over the nucleus, and then you have small foci formation, and this kind of makes the chromatin maybe less viscose somehow. I mean, it's a simple explanation. I'm not an expert in chromatin modulation, so I don't know whether this makes sense at all. Maybe somebody has a comment, but that's the only explanation that I can come up with.
Okay, well, I have some other questions for you, not from the panel, that I'll sort of start with, and then we'll ask both of you. I have some questions for both of you as well. How do you account for the gamma in the americium-241?
Well, it's negligible. We calculated it, and it's negligible. The impact is low, and also the energy of well, there is a spectrum of energy, but we calculated that some of them are just stopped by the Mylar foil. So basically, we decided to neglect it.
Another question: the increase of foci post-alpha irradiation may be in the non-irradiated bystander cells. Is that possible?
Yeah, sure, it is possible. We can't really tell. I mean, we have done some. We don't have a monobeam exposure facility, but we calculated. We did some Monte Carlo simulations. How many cells are actually hit, and how many cells show foci, and yeah, there are many more cells showing foci than are hit. So yes, of course.
But another comment, this is a broader comment on RBE. Is conservation for the sake of conservatism acceptable?
I think yes, we should be. I mean, we follow the precautionary principle. So we should be on the safe side, I think. So if we exaggerate, I mean, again, that's one of the questions that I ask is maybe is the radiation weighting factor 20 for alphas? Maybe it's sufficient. I mean, you seldom see, you really seldom get RBEs of 20. I actually don't know a single publication where you calculate an RBE of 20. Usually you get RBEs of around 5, 6, 10, maybe. So we are already kind of stretching, which is okay. I think that's the way to go.
I think there are some old neutron exposures that were up around 20 for the calculated RBE, but those were, those were done quite a while ago.
Yeah, and they look, and they are down on clonogenic cell survival. How valid is this?
Well, yeah, I totally agree. I'm not arguing that one bit. Use of cancer cells or immortalized cells is not ideal if it is for risk estimate purposes, and to make it more toward reality. Are there other cell types you could use?
Yes, well, that's why we do a lot of work on peripheral blood lymphocytes. I mean, the stuff on gene expression, but that I showed you, and chromosomal aberrations. This is all done on peripheral blood lymphocytes, but you can't culture them. I mean, it's difficult to culture them for a long time, so you can't really do chronic exposure experiments. Then you have to stimulate them with interleukins, and then you kind of introduce an artificial system anyway. Nothing's perfect.
Yeah, all in vitro systems are.
I know, Gail, that you like animal experiments. Yes, and I sure we need both.
Can you do yours in animals with your exposure facility?
No.
Yeah,
no, you can't really. I mean, we actually have submitted the project application to do mixed beam exposure facility with animals, where we will have to inject animals with some alpha emitters. And I don't know if I want this project to go through. We will see.
Okay, so what I'm going to do now is ask Laure to join us as well. There are a couple of questions for both of you, and then some questions that kind of go back and forth that either one of you can answer if you want. So Laure, if you'll kind of unmute yourself as well. Here's a question for both of you. Why did you choose to look at the chromosomes themselves instead of simply counting cell death as in earlier RBE studies? You alluded to that just a moment ago, Andrzej, but why don't you talk about it? And Laure, maybe you two can answer.
Yeah, in fact, we were really interested in chromosome aberration, in the fact that we want to look at which type of damage could be transmissible and could help induce in the cells some step toward carcinogenesis. And it's not dead cells who become a tumor, but it's a living cells. And it's a living cells with a lot of damage or a lot of modification. And we look at this first step of modification, which is a chromosome aberration. And that's also why we mainly focus on the type of labeling that could permit us to look at both unstable and stable chromosome aberration, to look at the chromosome aberration, to look at those that could be transmitted. And of course, I don't give you data about the chromosomal instability occurring in the progeny of irradiated cells, but that's what we had in mind when we start all those experiments. So we need to have a biological endpoint that will be an hallmark of carcinogenesis. And that's not the case for cell death. We have not in mind to calculate RBE when we perform the experiment or any RDE or anything else. So really the question was concerning carcinogenesis.
Yeah, I agree with Laure's answer. I mean, we are looking mainly at unstable type aberrations because it's easier, but to do fish, we don't. Yeah, I mean, I appreciate, Laure, your effort to do the fantastic fish stuff, but it takes so much time. But there is a good correlation usually between dicentrics and translocations. So think it's good enough.
Here's another question, probably more for Laure, but actually for both of you. There might be a large amount of interphase death after radiation. Are you missing this lethal effect by only comparing chromosome aberrations in viable cells? This would be true following carbon irradiation, but also, probably from the alpha that you're talking about as well, Andrzej.
For the carbon, with at 2 gray, we don't have this problem. We don't have a lot of differences in the mitotic index. But when we increase the dose, we really had this problem. And that's why I was not confident at 10 gray and 15 grays, because we have a huge amount of cell death.
Yeah,
and that is also a limitation to perform those effect curves because of the differences in the cell death.
Yeah, another problem is that cells exposed to high-LT radiation stop in the cell cycle. So that is why when we look at chromosomal aberrations, we always induce PCC with calyculin A, or a chemical PCC. But if you watched carefully, or if you watch the video, if you look at my slides, the dose response curve for chromosomal aberrations, they actually saturate at higher doses. And this is what we see repeatedly. And the reason for this is that we are exposing cells, lymphocytes, on these flat dishes. And this is very stressful for them. I think this is the reason. So we have some kind of overkill effect. Yeah, what can we do?
Okay, this is a question probably more for you, Andrzej, although both of you can answer. We should be careful about believing the linearity of biological values. These values may not be calculated like the way we calculate radiation dose values. One additional chromosome aberration to one existing aberration, one additional chromosome aberration added to one existing aberration, and one additional aberration that is added to 100 existing aberrations may not be the same.
Who should answer, Laure, or should I start?
I think it was meant for you, Andrzej,
yeah,
but
okay.
Certainly Laure can answer as well.
Again, I think that, yes, of course, I agree, but we are the experiments that we carry out, at the back of our mind is always radiological protection, and here we apply the precautionary principle. So we exaggerate. But that would be my answer. I really believe that we should assume linearity in cancer, radiogenic cancer risk, for the purpose of, or because we want to be on the safe side.
Okay. And Laure, do you want to say anything?
I have nothing to add from what Andrzej say. The only thing is that when we look at the chronic effect and we have cells who divide, when we add chromosome aberration to another one, it will not be the same for the transmission. So that, let's say it differently. When we have only one chromosome aberration in the cells, if the chromosome aberration is transmissible, the cells will divide, and you could have again living cells and chromosome aberrations that will increase. The total number will increase just because one gives two and so on. If you add chromosome aberration in a cell who already had chromosome aberration, the probability to have an unstable, of course, increase, and such the cells could die. So we will have a balance by the fact that we will have more induced death when we have this chronic exposure, and we have also more chromosome aberration due to the cell division. So it's really complex to analyze.
Great, thanks, Laure. I have a question probably more for you, Laure, than for Andrzej, although Andrzej can enter in as well. I believe Brenner and Shuriak proposed a similar quantity. They called it relative effects ratio, RER. For the data they were looking at, they found RER is less than RBE. Is there a sense yet how these RER or RDE quantities may vary across different endpoints and experimental systems?
I really think that this RD, it's different because, of course, we don't observe, we always observe that RDE was higher than the RBE. So I've not seen how Brenner has calculated the RER. Maybe it's the opposite way to calculate it. That's what we have done in RDE. So I need to check how he has done. But it's both the fact that any type of quantity vary across different endpoints is really due to the sensitivity of the endpoints that we are looking at, and also to how in what range the endpoint is comparable. I try to show you at my little level with the chromosome aberration. If we look only at an endpoint which is dicentric, we will miss a lot of information. If we look only translocation, we will miss a lot of information when we compare radiation quality that will induce different kind of chromosome damage. So that's why we try to look at the common point that they have, which is the chromosome breaks. So the fact that the quantity vary across different endpoints, it's maybe because the endpoints are different and that we are comparing endpoints which are not the same. And so we need to find a way to do it. And in experimental system, of course, they don't have the same sensitivity. And according to the sensitivity, we will have different way. And you could see when we have the, that's why also I think that we need to have a large number of donors to have a mean number. You have seen when Andrzej compares the patient 2 and the patient 4, he has different RBE because of the sensitivity, even if the endpoint is the same. So I think that to give an answer of the on such quantity, you need to have a very, very robust data, and we need to really be careful in the biological endpoint that we are looking at. Sorry for the very long answer.
No, thank you. Thanks, Laure. That was perfect. I want to end this soon because we were only supposed to go an hour, so we're obviously over time, but I think there are a lot of questions and it's been very interesting. So let me ask both of you a final question. Did you use the same dose rate for the different exposures?
Okay, in my case, the answer is no. There is, and the reason for this is that it's technical limitation. So the dose rate for X-rays is slightly lower than the dose rate for alpha particles, because I can't come close enough with the X-ray exposure facility to the cells. Yeah.
And Laure, what about you?
No, it's not the same one, because in term for the carbon we measure its influence, and it's not the same dose rate that we have, at least at the cellular level. The dose rate is absolutely different.
Okay, look, I think both of you have given the committee a lot of food for discussion. We're going to have to really work through how we're going to take these into consideration and how they're going to influence what we write. I suspect that we're going to be having some sections in the report that are going to have to deal with these very issues. And since Andrzej is on the committee, I'm hopeful that he will take the lead in some parts of that anyway.
I look forward to that.
We all do. And so let me give you, Laure and Andrzej, let me give you the chance to make any closing comments if you want to say anything at the end.
Just to thank you to ask me to exhumate all those data, and now I'm really excited to look at many of it.
Well, let us encourage you to go further. I mean, because this is cool stuff, really cool stuff.
Yeah. Same here. Thank you very much. And I mean, we are having 330 attendees. Wow, that's great.
Yes.
It's really great.
And let me tell you that I didn't read them, but in the comments, the Q&A, there were a lot of like, this was great, good job, a lot of really good comments. There's a lot of appreciation from the group for everything that you've done. We'll save the questions because some of them might be useful for discussion.
Absolutely.
Thank you all. Thanks to ICRP for hosting us. Kelsey did all the hard work of getting us all organized. And thank you to our fabulous speakers. This was just a really great session. Thank you. Bye,
Thank you
everybody.
very much.
Have a good day.
Bye-bye.
Bye-bye.